called the TATA box because it's a series of TATATAs. So the enzyme's going to bind to the TATA box, and you get elongation. Okay, adding the complementary RNAs. And then at the end you get termination. What happens in termination? RNA polymerase detaches. Okay, so the 3 steps. Whoops, this is initiation. Makes kind of sense, right? Initiation, you begin. Elongation, it grows. Termination, you end.
Yeah.
Okay. So let's say this is our pre-messenger RNA transcript. Okay, so this is going to be released. So you've got UCU, GUG, UAU, UCC. Okay, there's your messenger RNA. Once it's been released from the DNA, then you have what's called RNA processing. Okay, 3 steps to RNA processing: capping, tailing, and splicing. So first, let's just talk about capping and tailing first. A guanine cap gets added at the beginning. It's a modified G, gets added there. Tailing is at the other end, poly A tail. So a bunch of A's get added. Okay? And that serves a purpose, okay? Because number one, the messenger RNA has to get out of the nucleus into the cytoplasm. And once it gets out into the cytoplasm, there's all kinds of enzymes around that want to break down RNAs. And so, especially the poly A tail helps to protect the messenger RNA from getting broken down when it's in the cytoplasm. And the cap and the tail is also believed to help the ribosomes attach to the messenger RNA. Now, depending on how much of a protein the cell has to make, it's going to depend. The more protein you need, the longer the messenger RNAs need to be intact in the cytoplasm. So if you have you're going to make a lot of a protein and you're going to need the messenger RNAs for a while, the poly A tail is going to be longer. Okay, so the longer the tail is, the longer the lifetime of the messenger RNA.
Interesting.
Okay. So that gets them out. So that's another way of controlling the proteins that are being made. Okay, and then RNA splicing. This is something that's kind of interesting because you've probably heard that most of our DNA doesn't code for anything. It's what they call junk DNA. They don't call it junk DNA anymore. But if we were to look at a gene, here's a stretch of DNA that codes for a gene. Okay, this part here might code. Then you get a part of the sequence that does not code for anything. Some more coding. Not code. Coding. Not coding. Code. So what splicing does is it takes out these pieces of DNA that don't code for the protein. Okay? And so the ones that code are called exons. Okay, it comes from the term expressed sequence. Okay, it's going to be expressed as a protein. The ones that do not code are called introns. And that stands for intervening sequence. Instead of calling it junk, you call it an intervening sequence. So this would be exon number 1, exon number 2, exon number 3, and exon number 4.
So do we know why it puts those sequences that don't code for anything in?
Well, a lot of them have to do with viruses that have infected our ancestors in the past. I don't know if you talked about transposons, jumping genes. Okay, jumping genes can jump in there. Sometimes what happens is genes get duplicated. Okay, somehow during DNA replication you get copies of genes made, and they think this is the basis of evolution, those copied genes. And if they're not used for anything, they just sit there. But they think what happens, they think actually that's how our brains got so big. A certain gene for brain development got copied, and once it's copied, things can happen to it. Okay, it's not going to kill the organism to play around with it because it's still got a good copy. And so that's how our brains got so big. So anyway, there's a special structure that's pretty neat, but I don't think you're going to talk about it, called the spliceosome. It's going to splice out the introns. So in the end, this would be called your mature messenger RNA. It's going to be exon number 1, exon number 2, exon number 3, and exon number 4. So all the unnecessary information has been taken out. And one of the things that really shocked people when the Human Genome Project came out was how few genes we have. They would look, before they started it, they looked at the genes of a nematode, a microscopic worm, and said, well, we are this much more complex than a nematode. I mean, it didn't have a digestive system, it didn't have a circulatory system, it didn't have any of this stuff. So they were expecting about 100,000 human genes. When it turned out we had about the same number of genes as the nematode, people were absolutely shocked. Because we are just so much better. I mean, come on. But one of the reasons they think that we are able to get by with so few genes is called alternative splicing. So this one gene up here can be spliced to give you a different protein, just by putting the exons in different order. Okay, or maybe just keeping an exon out, you can get another protein, which is pretty cool. Okay, so everybody with me on this stuff?
Yeah.
Okay, now this part is hard to do on the board, but I do have some really good videos to show you.